weekly live educational series covering the COVID-19 crisis and the global community's efforts to develop a vaccine against the disease. Thank you so much to everyone who tuned in last week. We had a truly global audience with viewers from the US to India, Spain, and Brazil. It's such a reminder that what's happening is global. This is an unprecedented moment in history when the entire population, the entire world is engaged and just hungry to know as much information as possible from expert sources. And that's what this show is all about. We want to provide the most up-to-date scientific information so we can all better understand what's happening. Today, we have someone who 40 years ago worked with Dr. Tony Fauci to slow the spread of AIDS. We'll talk to her about the parallels between that fight and the one that we're in now. Then we'll go straight to the source. Dr. Paul Stoffels, the J&J scientist leading their vaccine efforts, will give us the latest on what's happening in their labs. And the United States has become the epicenter of COVID-19 cases. We all know this. We have surpassed all other countries in infections and deaths. One group of Americans is being disproportionately affected. And for more on that, we'll be talking to Dr. Cato Laurencin, who recently published the first peer-reviewed study on how COVID-19 is impacting the African-American community. If you have questions, please share them in the comment section, and I will be asking them throughout the show. We start with Dr. Peggy Hamburg. She's the former FDA commissioner and current foreign secretary at the National Academy of Medicine. As health commissioner for New York City in the 90s, she curbed the city's tuberculosis epidemic, and she has worn so many hats in the public health arena that I thought she'd be the best person to answer so many of the questions we have now. Welcome, Dr. Hamburg, to the show. I want to get right to it and take you back to 40 years ago. Even though I was quite young at the time, I will never forget the images of the AIDS epidemic. And in those early days, HIV, like COVID-19, was spreading among individuals who appeared to be asymptomatic. You worked with Dr. Fauci to help slow the spread of AIDS. How did you go about doing that? And are there lessons that can be learned and applied to COVID?
There are lessons to be learned, and I think it gives us reason for optimism. Certainly, no one expected the HIV epidemic to emerge. In truth, when I was a medical student, I learned that the era of infectious diseases was more or less behind us because of the advent of antibiotics and vaccines. And then we watched this mysterious disease emerge, taking the lives of previously healthy young adults. As I went through my medical training and I saw more and more the devastation of the disease, and we didn't have the medical tools to do much for these patients beyond just supportive care, I decided I really wanted to work on these issues. And that was when I had the fabulous opportunity to work as first Dr. Fauci's special assistant, and then as assistant director of the National Institute of Allergy and Infectious Diseases. And some really important things came out of that. One was that Dr. Fauci, Tony as we all call him now, really believed in going deep into the science, but also understanding the impacts on people. And he did something that was very unusual at that time, which was to reach out to those affected and really understand their disease experience and bring them to the table and work together, bringing industry, academia, the government, research and science community, NIH and FDA in particular, and the HIV affected community all together to develop a strategy for what needed to be done and then to implement. And it really has made a difference. And now what was always a lethal disease is a manageable chronic disease.
And do you feel like similar levels of collaboration is happening now?
Well, I do feel there are gaps in leadership, gaps in the ability to really galvanize the best and the brightest minds, the available science and technology, and the set of stakeholders and partners that are needed to really deliver on that promise of science. This is obviously a broader issue than just what was happening under the leadership of Dr. Fauci in the HIV research and product development arena, because it involves many other players. But that was true for HIV as well. And and thankfully, Dr. Fauci is still with us, playing that critical role. But we need, really, I think, a, a clearer strategy. We need to really define who is responsible for what. We need the federal government to step forward and take on more responsibility and accountability in critical areas. We need to define then the role of state and local governments, the private sector, and the broader communities, so that we can really make that necessary and urgent forward progress.
I said earlier that you have worn so many hats in the public health arena. I want to talk about your experience as health commissioner in New York, working in public health during an epidemic. In that case, it was tuberculosis. What were some of the defining factors and approaches to overcoming TB in New York City?
Yeah. Well, my experience with TB was shaped by what I'd learned with HIV, and I think is relevant now. Of course, the resurgence of tuberculosis that we faced in New York City was much smaller than what that city and so many others are grappling with now. But TB remains a worldwide public health challenge, with many people's lives lost around the world. And actually, Dr. Stoffels, who you'll hear from later, has been a great leader in the development of new products for TB. But a couple of critical lessons from our TB experience. One is, you need data. You need to understand what is the nature of the foe that you're dealing with. And for that, you need testing. We didn't realize we had such a big resurgence of tuberculosis back in the 1990s. And importantly, we didn't realize how many of the cases of tuberculosis that we had were actually drug resistant. So we weren't even treating them correctly. We weren't diagnosing adequately, and we weren't treating correctly. So some studies were done that allowed us to get a better sense of the problem before us, the contours of this unfolding epidemic. We then, under the leadership of then Mayor Dinkins, actually did what I was talking about. We brought together all of the different stakeholders across the city. We achieved a common level of understanding about the nature of the problem, and presented our strategic plan, which was very simple. It was diagnose the cases, make sure that the cases are properly treated, which with TB is hard because you have to treat on a regular basis over a prolonged period of time. So we had to actually send community workers out to find patients to make sure they were taking their medicine, and then to reduce some of the social settings that actually exacerbated the disease. So looking at prisons and jails, looking at congregate shelters, looking at hospitals and other congregate settings as well, so that we could reduce the conditions that contributed to ongoing transmission and spread. And with those three critical goals and objectives, an understanding of the responsibilities of all the different components of success, in terms of stakeholders and partners, we actually were able to turn the tide on TB far more quickly than we ever expected. And actually, our programs became a model for the nation and other places in the world as well.
Well, that's the thing about COVID-19. There is still so much that is unknown about this virus. Yet the federal government has been talking about this phase approach to returning to normal. But as you said, testing is crucial. So we've been hearing so many mixed messages. Where are we with testing? And are we where we need to be?
Yes, well, unfortunately, we did get off to a slow start with testing in this country, and we've been, I think, scrambling to catch up ever since. I'm hoping that we are at another inflection point where we will have more testing available. And it's not just having the diagnostic tests, but it's also having the tools you need to administer those tests, the swabs and the reagents for the tests. And there have been shortages of those. So we have to keep our eye on the importance of testing. Then we have to use that testing properly, both to make sure that all those who are infected, those with symptoms that are infected, uh, are tested, they're isolated so they won't continue to spread, and they importantly get the care that they need. Then we also do need to be doing some testing, what we sometimes call sentinel surveillance, so that we have a better sense of how the virus is penetrating into our communities, still perhaps silently, so that we can know where there may be new outbreaks of disease. So we really do need to get on the ball with the testing. And we are a country with enormous resources and huge scientific and technological capacity. We can and must do a better job, and I think we will.
Well, that's the thing, though. How reliable is the testing? There was recently contamination at the CDC. We've been hearing about how tests are sitting around too long and indicating false negatives. You look at a country like Korea that just they developed these tests and were able to disseminate them so widely. Why is it so hard in this country that you say is so developed and
Uh,
has so many resources?
Well, in all honesty, it has been a bit of a mystery to me why this has been so hard. Obviously, we're talking about a scale that is unprecedented, but it is such an important reminder when we talk about testing and diagnostics, but also when we talk about drugs and vaccines, that it really matters to evaluate whether the tests work. You've got to develop them and manufacture them, of course, but it's more than that. And people sometimes think that regulation, like the Food and Drug Administration, which I once had the privilege of being the commissioner of, that they just get in the way, that they're barriers to innovation. But the truth is that new products and innovation only matter if they make a difference for people. And if they don't work, if they don't do what they say they can do, then they don't improve people's health. They don't advance the health of populations. And in fact, people as individuals and the healthcare system don't have the tools that they actually need to combat a disease as devastating as COVID-19.
Dr. Hamburg, I have a question from a viewer on Facebook who asks, is the COVID-19 crisis phenotype similar to HIV in that it will change constantly and therefore developing one universal vaccine is wishful thinking?
Well, COVID-19 is a disease produced from a virus, as is HIV, but they're very different viruses. Another similarity is that they are both complex diseases that affect the body in many different ways and produce, as we learn more about them, different manifestations. In the early days of HIV, we thought we understood a set of symptoms associated with it, and then we learned that there were more and different ones. And the same is happening with COVID-19. It certainly is more than just a respiratory disease, and that creates new challenges in terms of how best to treat it. Um, I am more optimistic that we're going to have a vaccine for COVID-19 because of the nature of the virus and the opportunities to target certain portions of it to produce an antibody response and a robust immune response, and ultimately a vaccine. HIV, the challenge of developing a vaccine has been very elusive, but we've been very, very effective on the drug therapy side and have been able to, as I said, change it from a always fatal disease to a chronic manageable disease now through drugs that target the virus and its life cycle.
Well, speaking of vaccines, I want to take this opportunity to ask you about the process of FDA approval. You were the former FDA commissioner. That's the ultimate goal, right? To get your vaccine approved by the FDA. So what does that process entail, and how much collaboration happens between the FDA and the companies developing vaccines?
Yes. Well, vaccine development is usually a long and pretty complex process, and it's often a risky process as well, because at the end of the day, you're developing a product that you're going to give to people who are healthy to protect them from getting a disease. So the last thing you want is to have safety issues that make people worse. And of course, you want to have a product that will, in fact, protect them from that infectious disease threat. So ideally, as a company is starting to develop a vaccine, they begin to talk with the FDA about what the vaccine is and their research and development plans. And the FDA gives them feedback about what kinds of important questions need to be asked and answered in terms of their studies and the data collected. Generally, vaccine development happens in a very stepwise process, sort of assessing each step of the way, because companies don't want to make a lot of investments if they're going to fail. And you start with testing safety in a relatively small number of healthy people, and then you start to move it out into clinical settings where you get exposed, where people who are vaccinated get exposed to the infectious disease threat that, in the course of an ongoing outbreak or circulating infectious disease, and you assess the treated group compared to a control group to see if the vaccine is protective. And that can all take a long time and involve quite a number of subjects. In the case of COVID-19, because of the urgency of the problem and the magnitude of it, many of the steps are really sort of being collapsed, and people are willing, companies are willing to move more quickly. And even as many different candidate vaccines are now under development and going into human studies, people are already thinking about how are we going to scale up and manufacture in the volumes that are going to be necessary to address a global pandemic. So we're going in record speed from the time that the genome of this novel coronavirus was first posted in early January by Chinese scientists to the first injection of a vaccine candidate into a human. It was just a little over 9 weeks, which is an absolute record in terms of vaccine development. We're seeing rapid progress. Everyone has their fingers crossed. There's been unusual collaboration across all sectors and across borders. Really the mobilization of the scientific community to try to address this problem. Everyone recognizes that at the end of the day, it is a successful vaccine or vaccines that will really provide the blanket of protection that we need to make us go back to some kind of new normal again.
Well, we will be talking about vaccine manufacturing with Dr. Stoffels, but I just wanted to ask you, will the FDA approve more than one vaccine?
The FDA will approve any and all vaccines that are proven sufficiently safe and effective to make a difference against this devastating disease. I think the goal would be to have more than one vaccine. There are about 7 different broad categories of vaccine that are being developed using different strategies, approaches, and platforms. And there are, well over 70 different candidate vaccines that are in active development, and there probably will be more. So out of that pool, we all hope that there will be some options and that there may be some different strategies, and that we may also learn in the process of these vaccine development efforts more about new, better ways to make vaccines for the future as well.
Well, the FDA has a big job ahead of it. Dr. Hamburg, thank you so much for your expert insights. Now, in case you're just joining us, thank you. In case you're just joining us, I'm Lisa Ling, and this is The Road to a Vaccine, a weekly exploration of the COVID-19 crisis and the global community's efforts to develop a vaccine against the disease. I want to remind you to ask your questions in the comment section, and I will try to get to as many as possible. And now for Vaccines 101, which is a deeper dive into the basics of the vaccine process. Last week, we talked about what it takes to get a vaccine to clinic. Today, we're talking about how a vaccine gets manufactured. Earlier today, I went directly to the source, Dr. Paul Stoffels, who's in Belgium and is J&J's vice chairman and chief scientific officer, is going to tell us more about what's happening in the labs right now. So, Dr. Stoffels, you and your teams are working around the clock to get a potential COVID vaccine to patients as soon as possible, but you're also trying to upscale the manufacturing. Why is it important to do these things in parallel, particularly as it relates to this COVID vaccine?
Well, as we all know, COVID is global and affects all the people in the world. There is almost no region not affected. And that's where a massive upscaling is needed to, when there is a vaccine which works and it needs to be available to many, many people in the world, we need to have hundreds of millions, if not more than a few billion vaccines. And that's where upscaling and the preparation for upscaling is very, very important, that it started now to be ready when the vaccine is ready from the clinical trials.
So you start upscaling the manufacturing even before the vaccine is ready?
Well, we start the vaccine upscaling at the moment. Well, we only know that it works in animals. We have not tested it yet in humans. That will start in September. But in the meantime, we are already working on the preparation for very large quantities. That consists of two pieces. One is upscaling biologically. So we go from a 50 liter to 1,000 liter. We validate that. We do the quality on that. And that 1,000 liter vessel can give 150 million vaccines a year. And now we are also building more vessels in the world. So we build a new manufacturing plant in the US. We are working on more with partners in the rest of the world in order to get to a capacity to make more than 1 billion vaccines a year. And hopefully we'll be ready early next year to go to that level of 1 billion vaccines a year.
Have you ever produced this quantity of anything before?
No, in laboratory circumstances to test our technology, we have gone up to producing 300 million vaccines. That we have done already. So we know that we can do it. We have a cell line, what is called the Persis-6 cell line, which we have been developing for the last 10 years with our vaccine platform. And we know that we can make 300 million in a 2,000 liter capacity. So 2 times 1,000 liter, 2 times 150 million vaccines per plant per year. And that gives us the confidence that if we multiply the plants, then if we have 4 of those plants, we will be able to make more than 1 billion vaccines. Also in early testing, we have already in the laboratory demonstrated that we get to huge yields on our manufacturing cell line. And the combination of that makes us confident that we will be able to scale it up to very large quantities.
It's incredible. Can you describe the process of how vaccines are typically manufactured and what kinds of technologies do you use?
Vaccines used to be produced on eggs, so a very antiquated system, but still used today for several influenza vaccines. And then they use the eggs to grow the virus, and then the people host the virus and inactivate the virus. We now use cell lines for different types of vaccines. For example, vector-based vaccines, what we do where we use an adenovirus vector, it grows very well on a very particular cell line, the Persis-6 cell line, which we have developed for that. Protein type of vaccines, you can make them on cell lines. Some other vaccines are made in bacteria. So there are different platforms for different types of vaccines on how to get them produced. And all of them use different technology and different environments to be produced.
What would you say is special about your manufacturing technology?
Yeah, what we have been working on, because we were focused on pandemics before with Ebola, Zika, HIV, RSV, we have very particularly focused on an extreme high productivity cell line, which allows for extreme high output. And that's where we have been able to respond so fast to this epidemic and previous epidemics, that we have a cell line which can do that, as well as the combination with a vector, which together with the cell line forms, in fact, one technology. You can use both. If you use them together, you can make good vaccines as well as scale them up. And that's where we have a very particular technology to do this.
And what kind of capacity does that require?
Well, it requires if you would see the space, it's not that large, but if you see the technology, it's very complicated both from a technology perspective, but also from a biological perspective. This type of cell line and the manipulations you do to such a cell line is high tech. It's like one of the most advanced type of biological interventions you can do. Also, the development of a vector where you use a virus to carry another piece of the virus you want to make a vaccine against is one of the most advanced technologies you can use in the current environment. Yeah, and so it took probably 300-400 researchers 10 years to get to this point. We have a very extensive academic group, which with Dan Baruch, who has been collaborating with us. So as well, academic research. We have worked together with the U.S. military, with NIH, all in partnership to move this type of technology forward to enable to combat pandemics. We have focused on influenza because that's one of the most dangerous viruses next to corona, and so that gave us the experience and drove us to making and optimizing this technology.
There are a lot of people who are very concerned about vaccines to begin with. How can you ensure that your vaccine will be safe?
One, the vaccine is not a virus. It's a biological, it's dead. It's a biological carrier, but it's not a virus. So it will not replicate in your body. It will it will bring a piece of the virus into your body, and which is a small piece, which typically is not toxic, and it generates the antibody. Second, we have tested this vaccine technology in 4 or 5 different diseases already, and we vaccinated up to now, recently added another 10,000 people, so we have now 60,000 people who have been vaccinated with technology already, with minimal side effects. Overall, vaccines are probably the most effective way to prevent diseases. And I know very many people are scared from vaccines, but vaccines prevent today many diseases from your childhood, measles, mumps, rubella, hepatitis now, all types of diseases are out of our life because of a vaccine. And so vaccine has been one of the most important technologies to bring the life to where we are today, only in eliminating diseases from our society. Yeah, today there is no polio anymore. Where is that? Why is that? Because we had a vaccine. There are no cowpox anymore because we have a vaccine. So it's all thanks to vaccine technology that we are who we are today and can live healthy lives. And we know now that we need many more vaccines to protect ourselves to next diseases, now like COVID or Zika or other diseases which appear out of the blue.
And that was Dr. Paul Stoffels, J&J's Chief Scientific Officer, with whom I spoke earlier. For those of you just joining, I'm Lisa Ling, and we're back with you live. I want to encourage you all to ask questions in the comment section, and we will be answering as many as we can. Our next segment explores what I think is one of the most concerning and devastating aspects of the crisis, how COVID-19 is impacting Black and Brown communities in this country. My next guest is Dr. Cato Laurencin from the University of Connecticut. He's a professor and surgeon, and he recently published a study that found significant differences in the way COVID-19 is impacting communities of color. Dr. Laurencin, thank you so much for being with us. What is your perspective on COVID as a surgeon, engineer, and physician in the community, and how has COVID impacted you?
Well, Lisa, thank you. First, I want to thank you for having me on. I do want to take a moment to thank all the first responders and for what they've been doing, especially at the University of Connecticut. They have been truly heroes, from those working in the emergency department to those working in the ICU, but also the staff of the police, janitors, other essential employees. They really have shown their true colors in their work, and so I think that we need to have a tribute to them. The COVID-19 pandemic has had really a devastating effect in terms of the Black community, and we sort of, people have asked me, how did this all start in terms of our writing our first paper in this area, And it really came about in March when I looked on the internet. And if I place the keywords COVID-19 and Blacks, I found that most of the internet passages were, can Blacks get the coronavirus? Are Blacks immune to the coronavirus? And this was really very surprising to me that that would be on the internet at that point, but also frightening to me because that type of misinformation in the Black community could be deadly. And that's what really prompted our moving forward to get the information and to write the first paper on COVID-19 and the Black community, looking at real data on the Black community. Interestingly enough, we went to the state of Connecticut Department of Public Health to talk to them about getting the data, and the data was not available on the website. And we actually had to go through a number of different steps to be able to obtain the data, including signing release forms, et cetera, to be able to obtain the data. When we found the data, when the data was brought to us, we were equally shocked that only a percentage of individuals who were tested had race and ethnicity data that were logged. And this was, again, extremely concerning. And we've actually found across the country that race and ethnicity data is actually very, very low. And so the pandemic has really specifically and affected the Black community in ways that are far-reaching and I think ways that we'll be seeing, for years and maybe even generations to come.
And so you published a study about COVID's impact on communities of color. What were your findings?
Well, our findings were that compared to the general population, to the overall population, that Blacks had higher rates of coronavirus infection and also higher rates of coronavirus deaths. Now, this is in the context of the internet, in which, at the time in which we were putting together the study, had placed questions as to whether Blacks were immune. In fact, in the state of Connecticut, death rates per 100,000 of Blacks versus whites are twice as high for Blacks and whites, and coronavirus infection rates are 30% higher in Blacks than whites. We're seeing this, of course, across the country now. It's been reported after our paper, it's been reported across the country that other centers and other sites and other hotspots are having these sorts of rates.
So it was actually the opposite, your findings of what you initially saw on the internet, that Blacks had immunity. Did you expect to find the results that you did in your study?
We did, and that's what prompted us to try to dispel the myth and also to address the misinformation that was taking place in the Black community. We know that there are a number of contributing factors that take place in the Black community, making individuals more susceptible to disease and death by coronavirus. And these are all under the general area of discrimination and racism that's taken place over hundreds of years in terms of America and continue today. And so we know that in terms of areas of comorbidities, in terms of hypertension, asthma, COPD, diabetes, these rates are higher. And there are a lot of factors that are even behind those. One compares Blacks versus whites in terms of health insurance. Blacks are twice more likely to not have health insurance. Have higher rates of situations in which a doctor visit doesn't occur because of cost. And so we know that in terms of housing, the history of housing discrimination has taken place in America has been going on for over 100 years. And in fact, the last anti-housing discrimination laws were actually passed in the 1990s, if you believe it, 1990s. And so in terms of housing that has areas such as food and proper food, proper groceries, proper medical supplies, these are low in the Black community. And also, of course, we know that in terms of the concept of the essential worker in the work that can't be done at home. There are higher rates of blacks who cannot work at home and cannot be sheltered in place, and are working in outside the home during this period of time, and working in hazardous positions. I saw one quote that 60% of nurses aides in nursing homes, which of course there are hot spots in and of themselves. 60% of nurses aides working in nursing homes are Black, and so this provides another level of concern. And of course, with the misinformation about coronavirus, and that misinformation goes on even now in terms of what the proper protection is and whether one should shelter in place or not shelter in place. It creates an environment in which it can be an environment for disaster for the Black community.
You're talking about some of these just devastating systematic challenges in communities of color, but from a health perspective, why are African Americans at a higher risk to severe infection with COVID?
Well, again, I think that one is, of course, is that this is a disease that feeds upon comorbidities, and those comorbidities are there. It's a disease that is highly contagious, and so those situations in which one is in an environment where one can contract the disease, and Blacks are placed in that environment, whether one's a bus driver, janitor, one's in one of these essential positions in which one has to be present, places individuals in locations that these things that these diseases can be contracted. And so it's very, very important. And I guess the third, of course, is that what we see now in the state of Connecticut, for instance, I think it was quoted that 8% of individuals who are white they test positive. Over 30% of individuals who are Black are testing positive. And it's what we know is that the level of testing in the Black community that's taking place is not sufficient. And we still see the institutional discrimination still taking place, where Blacks are not being adequately tested, and when they are tested, they're actually they're found to have coronavirus at a much higher rate. So these are all contributing factors that take place that actually creates an environment where coronavirus is high in the Black community.
Dr. Laurencin, I have a question from Michelle from LinkedIn who asks, you mentioned a lack of demographic data. How did you get reliable data to draw such confident conclusions?
Well, the first is that we were persistent in there are three answers. One is that we were persistent in demanding. And so I actually spoke directly with the chief medical examiner for the state of Connecticut and asked for the data and said and made it very clear how important it was. Some of the data didn't have race and ethnicity. I asked that we get that data in terms of that, and in fact, for race and ethnicity, for deaths from coronavirus in the state, we actually have a fairly high percentage. But the other part of the answer is that for the cases, we still have issues in terms of data on coronavirus cases by race and ethnicity, and it's really all over the country. And we really, in terms of one of the action points, we must demand that we have adequate data on race and ethnicity. We know that diseases such as HIV that start everywhere and start in a number of different communities end up being concentrated in the black and brown communities. In fact, over 60% of all new HIV cases each year are in Blacks and Latinos right now. So we know that this is what happens in the Black and brown communities, and so having that data is important. The last segment talked about HIV and some parallels. There are parallels. In 1981, the first paper on HIV came out. It wasn't until 2 years later that race and ethnicity data was reported for HIV, and we lost valuable time understanding the changes that are taking place in terms of HIV moving from predominantly a white community to the Black community. And so we don't want to make the same type of mistake again. And so I think having adequate data to track it now and to track it in the future is very important.
Rebecca from LinkedIn is asking a question that I was about to ask you. your study was conducted in the United States, but do you believe there would be a variation in these ethnic disparities overseas? Is it happening overseas as well? For example, in the UK or other parts of Europe?
Yes, what we're seeing is that these same types of disparities are taking place in the UK. And in the UK, the group is called Blacks and Other Minorities, and a study has been recently published on Blacks and other minorities, demonstrating that even though, again, the percentage compared to the percentage of Blacks and other minorities in the country, the percentage of Blacks that are and other minorities that are contracting the coronavirus is very, very high. Interestingly, when we talk about the fact that they examine the numbers of individuals, a major point was the first 9 or 10 individuals in the UK who died of coronavirus in terms of health workers were all minority. I know we have to wrap up soon, but I just want to make up one or two important points. The first is that I believe we have to do things a bit differently than we've been doing it. If we look at what's going on right now, we're now going to be passing 1 million cases of coronavirus in the country, over 50,000 dead. We need to create systems in which we examine and diagnose people earlier. There's a famous study in Shenzhen, China, in which they actually looked at the tracer work and found 55 people who were asymptomatic with coronavirus. They did chest CTs on all these individuals and found 1/3 had pneumonias, even though they were asymptomatic. And they advocated early treatment. And when they did, people got out of the hospital setting very early, and none went on ventilators. In New York City, over 90% of individuals who go on a ventilator actually die. And so we need to do everything we can to avoid individuals going onto a ventilator. And we need to, especially if there's capacity, we need to have early admissions, even with people who are asymptomatic or minimally symptomatic, to allow individuals to be able to be treated and to avoid the complications taking place.
And Dr. Laurencin, I just have to ask you, do you have any solutions or advice for communities that may be more susceptible about how to proactively seek help?
Well, I think there are three things. One is that I think it's very, very important that information flow better. And while having this information is great in terms of going out, a number of individuals in the Black community won't be watching this. And so we have to have directed, targeted information provided to Black communities in terms of the coronavirus. And number two is that nationally, we've got to decide where we're going in terms of the coronavirus. On one hand, we've got shelter in place. On the other hand, we've got the beaches are open. And so we've got to really have a real national policy in terms of how we're going to be able to combat the coronavirus and move forward and make sure that's in the community. Third is that we need to have adequate testing. The testing in the Black community is very, very poor, and we need to make sure that testing is available to those in the Black community. Remember, many of the ways in which one gets tested, going to their primary care doctor with symptoms and getting a slip to get tested, it won't occur in the Black community with lower amounts of access to healthcare, lower amounts of insurance that's there. So we have to create solutions. And then lastly, as I mentioned earlier, I think we have to do things a bit differently. We have to do tracer technology. You have to use tracer technology to trace those that the first degrees of those after infection. We have to provide early treatment. Early treatment is so important, even with a pop-up tent hospitals, with early treatment to be able to prevent the types of complications that we're seeing from coronavirus and the death rates that we're seeing in the Black community.
Dr. Laurencin, thank you so much. It's so important to have your voice in this. And I want to let you know, I've been told that J&J is working on some treatment options for the population you've been studying, and we'll be discussing more of that on this show and future episodes. So keep submitting your questions at jnj.com/road to a vaccine, and we'll be answering more on Tuesday next week live on another episode. Now, it's often the most difficult times that bring out the best in us. So I wanted to highlight a few everyday heroes who are making a difference in their communities. So before we go, check out the short video, and thanks so much to our guests and to everyone for watching.
It was the absolute least I can do to show my appreciation and help spread some sweetness during this time.
We really wanted to get involved to give back to the people that are putting their lives at risk for us.
We are in this together. Thank you.
The fact that there's so many hardworking people just going to work, we wanted to be sure to show them how much we really appreciated them doing everything they can.